There are several differences between mitosis and meiosis, which include number of divisions, presence of the synapses, number of daughter cells and genetic composition, and the structure and movement of chromosomes during anaphase. The main importance of mitosis is maintaining the chromosomal number of the cell in order for the cell to complete the cell cycle and enter meiosis. Mitosis is also very important for reproduction, growth and development, and tissue renewal. Without mitosis, cells could not replicate and would cease to exist.
90% of the cell cycle occurs during interphase, which is divided into three subphases. The first phase is G1, which is when the cell grows in size and mRNA and proteins are synthesized in preparation for mitosis. Next is S phase, where DNA is synthesized. And lastly is G2, where the nuclear envelope encloses the nucleus and two centrosomes are formed.
In prophase, the chromatin fibers start to condense and each duplicated chromosome appears as two identical sister chromatids joined at their centromeres. Also, the early mitotic spindle starts to form and the centrosomes begin to move away from each other due to the growing microtubules extending from them. By this phase, the centrosomes are at opposite poles of the cell. After the attachment of kinetochore and microtubules to the kinetochores of the sister chromatid in prometaphase, they convene at the metaphase plate, which is a plane that's equidistant between the spindle's two poles. Anaphase is the shortest stage of mitosis. In this phase, the sister chromatids start to separate, and each chromatid becomes a chromosome. The two free daughter chromosomes start to move toward opposite ends of the cell as their kinetochore microtubules shorten. The cell elongates due to the lengthening non-kinetochore tubules. By the end of this phase, two ends of the cell have equivalent and complete collections of chromosomes. During telophase, two daughter nuclei form in the cell and the nuclear envelope begins to appear, along with the nucleoli. The chromosomes become less condensed and any remaining spindle microtubules are depolymerized. Also, the cytoplasm is divided in two and the two daughter cells begin to form. In cytokinesis, the cleavage furrow starts to form and pinches the cell in two to create two complete daughter cells genetically identical to the parent cell. The result of mitosis is a separation of sister chromatid to form two diploid cells that are genetically identical to the parent cell. In meiosis I, homologous chromosomes are separated, diploid cells are transformed into haploid cells, and two cells are produced. In meiosis II, sister chromatids are separated, and four haploid cells are produced.
During prophase I, homologous chromosomes coil and become shorter and thicker. The paired homologs become connected, which is known as synapses, and during this process, crossing over occurs, which means there is a rearrangement of genetics between pairs. During metaphase I, the chromosomes line up on the metaphase plate. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. During anaphase I, the breaking down of proteins allows the homologs to separate. The kinetochore microtubules shorten and pull the sister chromatid toward the poles. During telophase I, each half of the cell has a complete haploid set of duplicated chromosomes. Cytokinesis is the division of the cytoplasm where each daughter cell receives an equal amount. Both telophase and cytokinesis occur simultaneously. During prophase II, a spindle apparatus formed. Unlike prophase I, synapsis does not occur. During metaphase II, the chromosomes are positioned at the metaphase plate as in mitosis, but now because of crossing over in meiosis I, the two sister chromatids of each chromosome are not genetically identical. During anaphase II, the proteins holding the sister chromatids are being broken down and allow the chromatids to separate. Finally, during telophase II, nuclei form, the chromosomes begin decondensing, and cytokinesis occurs. The end result of meiosis are four genetically unique haploid gametes with 23 chromosomes in each. So why is meiosis important? Well, it creates genetic variations among a population. Also, it produces haploid gametes. Without meiosis, life will cease to exist.